Well, thank you, and good morning, everyone. I appreciate you taking the time to come and give me the opportunity to introduce Lisata Therapeutics to you all. Like all the other companies that are publicly listed today, I'll just take a moment to remind you I'll be making some forward-looking statements. And I ask that you please keep those in mind if you take investment decisions based on what you hear today. So let me start with a brief overview of Lisata, which is actually a company that was formed in September of 2022 based on the acquisition of CendR Therapeutics by a company called Caladrius Biosciences, which was our predecessor company. And we went through that transition specifically because of the technology that you're going to hear about today, and are now focused entirely on developing and advancing this technology for the treatment of solid tumors. It's a very novel targeting and penetration technology that is designed to improve the efficacy of concomitantly administered anti-cancer drugs. And like many companies, we have a very seasoned management team. Of course, our intellectual property portfolio is quite solid. We have patent protection, which includes composition of matter, method of use, and a variety of other important applications through most of the next decade, and in some cases into 2040s. Importantly for investors, we actually will be reporting a large number, about a dozen or so important data milestones over the course of the next 12 to 18 months. You'll hear more about that in a moment. And we are fortunate that we have a number of consummated R&D and commercial deals, which we believe also lends credence to the technology's potential. But maybe most important is the fact in these difficult times, we are actually well funded. We have cash that will take us into the early part of 2026, which is sufficient capital to fund all of the programs about which you will hear through to data. And so we're currently not actually raising money, but like biotechs everywhere, if someone was interested in making an investment, we'd certainly like to talk to them. So I won't spend too much time on the next two slides, just to point out that our management team has almost 2 centuries of collective development experience, combining big pharma, small pharma, and service provider experience. We also have collectively contributed to approximately 75 drug approvals internationally, many of them in the oncology space. And we have a similarly accomplished industry veteran board of directors who are all pharma experts. And so that really helps guide our company well. So why are we in oncology? Well, I think most people understand that cancer remains a significant unmet medical need generally around the world. But what's not typically known is that about 90% of the newly diagnosed cancers are in fact solid tumors. And these solid tumors include all those that you see on the bottom of the slide, those that have very poor prognoses and where the outcomes for patients remains poor. And the reason for that is that the existing treatments are really suboptimal. And there are a number of reasons why that's the case, but principally it's because of the morphology of these solid tumors, their structure. They create a physical barrier called the stroma, which consists of a variety of different types of cells, including tumor cells and tumor endothelial cells, which acts as a physical barrier preventing the infiltration of anti-cancer agents. And they have an immunosuppressive tumor microenvironment, which really hides the tumor from your immune system and also minimizes the effect of externally administered immunotherapies. And you really can't get around this by brute force, which has been the typical approach in the past. You just give higher doses, try to force things through by a concentration gradient until you get off-target side effects, which often are intolerable and cause the patients to stop the treatment. So we have found a technology that really is the combination of 2 very distinct breakthroughs in the understanding of biochemical mechanisms. On the one hand, a mechanism that has identified RGD motif peptides as being very good targeting agents for solid tumors based on certain receptor affinities. And then the next step of that, which finding a way to modify those to make those internalizing RGDs, which are in fact RGD peptides which commandeer or hijack a naturally occurring active transport mechanism to transport products into the solid tumor itself. And our lead compound, certepetide, is in fact an internalizing RGD peptide that's in mid to late stage clinical development. And certepetide works by converting the tumor stroma from a barrier to an actual conduit. It also modulates significantly the tumor microenvironment, selectively depleting immunosuppressive T cells, enhancing the concentration of cytotoxic T cells, and actually inhibiting the metastatic cascades. This is very, very important. And as a monotherapy, it does those things, but we're developing this compound to be used in combination with other therapies. And what makes this so interesting is that the mechanism of action of certepetide is agnostic to the mechanisms or the modality of the concomitantly administered companion drug. So we can use this with chemotherapy, radiotherapy, immunotherapy, antisense therapy, even cell therapy, and we get the same type of result, which is really very important. As a result of this, we've developed a two-pillar strategy for the development of this platform. And it's based on the fact that these solid tumors, especially the GI solid tumors, are becoming more and more prevalent. And for example, by the end of this decade, pancreatic cancer is expected to be the second leading cause of cancer death, and it remains very poorly treated. So the first pillar here is to pursue a rapid global registration in metastatic ductal adenocarcinoma, initially combined with one of the two standards of care in this area, which is gemcitabine, nab-paclitaxel. And in that side of the strategy, we've actually completed the enrollment of a large phase 2b, and we'll have data readout from that toward the end of this year, which will be seminal for the company. On the other side, we're looking to exploit fully the broad applicability of certepetide in combination with many other anti-cancer regimens in a variety of different solid tumors. And we have a pretty rich portfolio of early stage phase 2A type programs looking to demonstrate proof of concept in that regard. I mentioned earlier that we have a number of partnerships. On the one hand, we have R&D alliances, which provide us with resources and in some cases foundation money, non-dilutive capital, but they take no commercial interest in the product. And then we do have a strategic commercial partnership with Qilu Pharmaceutical in China for the development of the product in the Greater China territory. And we've been collecting significant milestones and have a number of additional significant milestones to collect from Qilu as the development continues forward. But because of the ability to mix and match certepetide with so many things, we can envision a wide variety of non-exclusive type partnerships by indication, by modality of the co-administered drugs, and or by geography. So really a very attractive commercial opportunity that complements the attractive medical opportunity. So it's probably important to tell you how this works. If that's mine, I apologize. But certepetide is a 9 amino acid cyclic peptide, and it has a very high binding affinity and selectivity for certain integrin receptors, alpha B beta 3 and alpha V beta 5, which are found on the componentry of the stroma. And so once it's bound to these integrins, it becomes susceptible to proteolytic cleavage, and it releases two linear peptides, one of which is almost a nutritional supplement, if you will, 4 amino acids that just get absorbed into the body. But the 5 amino acid fragment is actually a CendR rule or CendR fragment, which has a high affinity and selectivity for binding to neuropilin-1, which is found in the same tumor microenvironment. And once neuropilin-1 is activated, it actuates the CendR rule active transport mechanism, and that's manifested by the formation of microvesicles on the surface of the cells, which encapsulate the co-administered drugs and percolates or ferries them through the stroma and deposits them deep into the tumor. And at the same time, certepetide has the effect of modulating the tumor microenvironment, as we discussed, making those cold tumors hot and making them more susceptible to recognition by your innate immune system, as well as perhaps more susceptible to externally administered immunotherapy. The next couple of slides just simply show how selective and efficacious this is. And this is a preclinical experiment where the co-administered agent is really a fluorescent quantum dot. And in these experiments, when you administer the FQD and you apply a quenching solution, any of the FQDs that would be in circulation would go dark. And so you have a dark mouse there because everything's in circulation. If we add certepetide to the same experiment, you see all the FQDs go into the tumor model. And you have very selective penetration of that pancreatic tumor model here. And it shows that you have very targeted penetration. And then there are almost 360 now scientific publications that demonstrate consistently that when you add certepetide to a combination of other anti-cancer agents in any validated solid tumor model, what you see is an improvement in efficacy in those other agents. And so that's really something that has been consistently shown in vivo and in vitro in animals. But of course, what's most important is what happens in the animal of interest, which is people. And these are the results of our multi-center phase 1b, 2a trial done in Australia in pancreatic cancer patients, metastatic and first-line patients. And of course, as a first-in-man study, first thing we looked at was safety and tolerance. And not surprisingly, certepetide was well tolerated. We cannot find a dose-limiting toxicity for this agent. And the safety of the combination of certepetide and standard of care, chemotherapy in this case, was consistent with the standard of care chemotherapy alone. So we did not exacerbate at all the adverse events. But what we did augment considerably were the efficacy effects of that chemotherapy. And you can see that, I think, a little better visually here, where you see a 55% improvement in overall survival and a 76% improvement in median progression-free survival. These were identified as breakthrough type improvements by the investigators involved in this study. Additionally, what was very important and intriguing for us was the fact that all of the other efficacy markers consistently showed an improvement. So in clinical studies, I think you all know what you want to see is all of the markers moving in the same direction. And there's nothing confounding here. So again, it gives us great promise that things could go forward here, not only in metastatic cancer, but perhaps generally in other solid tumors. And recently we had another indication. Now this is an N of 1, so we all take this as such. But this is an important, I think, experiment because it shows that in metastatic gastroesophageal cancer, where a patient who was really at the end of their treatment options, they had received radiotherapy and chemotherapy with added immunotherapy and still were not seeing an adequate response. When certepetide was added to their treatment regime after several more cycles, they actually showed a complete response. So again, further evidence that we can mix and match this agent with a variety of different things and expect to see significant improvement in the outcomes for patients. We have also the validation, if you will, of that from the regulatory agencies, where we have received fast track designation from the FDA for pancreatic cancer, orphan drug designation in pancreatic cancer from the FDA and the EMA, as well as orphan drug for malignant glioma and for osteosarcoma. We also have a rare pediatric disease designation, and as I think you all well know, these carry more than just regulatory benefits, but they actually ultimately have commercial benefits because they provide for the opportunity for accelerated approvals, for the access to grants, tax credits, and importantly, market exclusivity. So the next several slides show the pipeline of products we're developing. Probably the biggest one is the first line, the ASCEND trial, 158 patients, double-blind, randomized, placebo-controlled, and powered for efficacy to demonstrate certepetide's addition to gemcitabine nab-paclitaxel will increase the efficacy of that regime without hurting the safety profile. And that data is expected by the end of this year, and that will be seminal. But in addition, we have a number of other trials running, and I'm not going to read the slide to you, but you can see a variety of solid tumor types in combination with chemotherapy plus immunotherapy, and several others, including trials in glioblastoma, and of course the trials running by our partner in China. And so when you look at the milestones over the course of the next year or so, you can see, and this is just the clinical side, you have the circles are data execution milestones, the stars are data reporting milestones. You'll see a steady news flow, a cascade of news coming from all of these trials, starting toward the end of this year and continuing out through 2025, any one of which should be value creating, but as a accumulation should really substantially increase the value of the compound and increase the interest that we have in the products going forward and the company. So financially, I mentioned we have about $43 million as of the end of the first quarter of this year. We burn on the average about $5 million a quarter, but that's a decreasing amount as we finish enrollment in a number of these other trials. So easily have cash into the first quarter of 2026, probably longer. Our finance group is very conservative, as you would like them to be, but I'm sure we'll be able to stretch that beyond should we need to. Our cap table is very clean, and most importantly, we carry no debt whatsoever. So in the end, we think that the company represents a compelling investment thesis. We have a very promising asset that's based on a body of compelling preclinical and now clinical data with accumulation of information that is generally indicating that everything continues to move in the positive direction, supporting the hypothesis that certepetide improves the efficacy of co-administered agents without exacerbating the adverse events. We have a very rational, focused, and very capital efficient development program. You can see we're running about a dozen different clinical trials. For a small company, that's a lot, and still keeping our burn very, very under control. We're only 27 people within the company. Very highly experienced management team, and of course, a fiscally stable company. And this is just a repetition of what I just said, but I think you all know that when you're talking to an audience, you're supposed to tell them what you're going to tell them, then you tell them, and then you tell them what you told them. And so that's what I told you, okay? And I hope that you find it interesting. And if you're interested, I would love to see you become shareholders of our company and join us on this journey to improve the treatment of solid tumors in patients. And thank you for your interest, and I have a few minutes for questions. Thank you.